Hello everyone, welcome to the physiology class of the Department of Special Education and Rehabilitation Technology. Do you know what the most numerous organ in the human body is? Do you think my question is a bit odd? You must be thinking, the organs in our body are either single, such as the heart, liver, and spleen, or paired, such as the kidneys, eyes, and ears. How can there be a most numerous one? Let me reveal the answer to everyone. The answer is muscles. Because each individual muscle is an organ, the human body has more than 600 muscles, which are widely distributed, accounting for approximately 40% of body weight. Each muscle has a specific shape, structure, and auxiliary structures. It is supplied with abundant blood vessels and lymphatic vessels, is innervated by nerves, and can perform complete contraction and relaxation functions. All forms of movement in the human body are mainly accomplished through the contraction of skeletal muscles. Based on differences in structure and contraction characteristics, human muscle tissue can be divided into three types: skeletal muscle, cardiac muscle, and smooth muscle. Among them, skeletal muscle and cardiac muscle, under a light microscope, show alternating light and dark striations, so they are collectively called striated muscles. In addition, based on differences in neural innervation and control, muscle tissue can also be divided into voluntary muscles and involuntary muscles. Voluntary muscles are controlled by our consciousness, just like how we are sitting here in class now. We are consciously maintaining our sitting posture. Therefore, most skeletal muscles are voluntary muscles. How does our brain control our muscles? To understand how nerves control the movement of skeletal muscles, we first need to understand how nerve cells and skeletal muscle cells connect. The skeletal muscle neuromuscular junction is a specialized structure between the motor nerve terminal and the skeletal muscle cell it innervates. It is composed of a presynaptic membrane, a postsynaptic membrane, and a synaptic cleft. The presynaptic membrane is a part of the membrane at the end of the motor nerve axon. The postsynaptic membrane is the skeletal muscle cell membrane opposite the presynaptic membrane, also known as the end plate membrane, and has the shape of a shallow, inwardly concave groove. The synaptic cleft is the space between the presynaptic and postsynaptic membranes, with a width of approximately 20-30 nanometers, nm, and is filled with extracellular fluid. In the axoplasm inside the presynaptic membrane, there are about 30,000 synaptic vesicles, also called synaptic vesicles. Inside each vesicle, there are approximately 10,000 acetylcholine, ACH, molecules. In the postsynaptic membrane, there are type N, ACH receptor cation channels. On the outer surface of the postsynaptic membrane, acetylcholinesterase is also distributed. This enzyme can break down ACH into choline and acetic acid. The process of excitation transmission at the neuromuscular junction has the characteristic of electrical transmission. Specifically, the action potential transmitted from the motor nerve fiber to its terminal is the electrical signal. It triggers the Ca-dependent exocytosis of synaptic vesicles from the presynaptic membrane, releasing ACH into the synaptic cleft. This is the chemical signal. Then ACH activates the type N, ACH receptor cation channels in the end plate membrane, generating a potential change in the end plate membrane. This returns to an electrical signal. Type N, ACH receptor cation channels mainly allow Na influx. The net influx of Na causes depolarization of the end plate membrane, and this potential change is called the end plate potential, or EPP for short, from end plate potential, with an amplitude of up to 50-75 millivolts MV. EPP is a type of local potential. Within a few milliseconds after ACH is released, ACH is rapidly broken down by the acetylcholinesterase on the outer side of the end plate membrane, thus eliminating its effect. This allows the end plate membrane to return to a state where it can receive new excitation transmission.